Greetings, ladies and gentlemen, and welcome once again to the Compendium of Mad Science. I'm afraid I've been off with the flu for a little bit, so my voice is a little bit croaky. Hope you don't mind, but I've been really wanting to get this video out for a while now. It's also really cold in here. I'm not sure if that's a remnant of the flu, or if I've cut myself while shaving. I'm not sure if that's a remnant of the flu, or if I'm just at a funny age. Either way, I'm not feeling very warm, so I'm going to button my lab coat right up. There we go. Right. Welcome to episode 3 of the Compendium of Mad Science. Today is the first of our new segment entitled Microscopy Monday, where we do, wait for it to, this is going to surprise you, we do microscopy on a Monday. Today's actually a Saturday, but I'm going to release it on a Monday, and that's what's important, the release day. Right, so we're going to go through the absolute basics of microscopy. We're going to start at the very simplest aspects. We're going to look at the microscope, tell you what all the parts do, then we're going to go through a simple slide preparation, and then finally we're going to look at some slides. If you're already familiar with microscopy, I advise you skip right on to the end, because you're going to find it boring when I tell you what a focus knob does. I'm sure you already know that. I also sure you know what a wet and dry mount is. I don't expect everyone to know that though, so this is aimed at the people who have no idea about microscopy. The idea of this is to get you started, enough to get you doing basic microscopy in your house for a relatively low cost and with moderately little preparation. In future episodes we'll cover more advanced techniques like staining, actual histology, and different types of microscopy. I mean, I've only got a light microscope here, but you can do transmission and reflective light microscopy with that. This is aimed at absolute beginners in microscopy, or at least the first part of it is. Again, if you already know about microscopy, feel free to skip ahead, I won't blame you. Well, I've got a couple of samples here for us to look at today, so I'm going to put on some gloves since I know at least one of them came from a river, and I don't know what was in that river, but I'm pretty sure it was polluted, so I don't know what bacteria or fungus is growing in it. I also deliberately took it from a fungusy area, so almost certain that there is something interesting growing in it, but hey-ho. As long as you take precautions, everything should be fine. First precaution I'm going to take is making sure I've got stuff ready to disinfect other stuff. This is just Dettol solution. I'm going to get a bit more water in there. I'm using Dettol for the simple reason that it's cheap. I would ideally use alcohol, preferably isopropyl alcohol in a fairly high concentration, but I don't have any kicking about just now. What, an electronics lab that doesn't have alcohol? Yeah, I know. I'm ill prepared. Right, so this here is a basic light microscope. As you can see, I've actually got a digital sensor plugged into it just now, so I'm going to change that out, because we don't want a digital sensor when we're going through the basics. There we go. Right, so this is a very basic light microscope. This particular one is made by Brunel Microscopes Limited, but you can get any microscope you want. You can pick them up for, you can get them for as little as £10 or £20 nowadays. You do not need terribly expensive equipment, though if you'd like to send me a terribly expensive microscope, I would not say no. So this is a light microscope. As I said, this one's been for quite a while now, since I was about 15. And it certainly does me fine for the sort of stuff that I tend to do in the lab. I'm just going to take you through the absolute basics of the light microscope. This is what's technically known as a transmission light microscope, which means you put your sample here and the light is transmitted through the sample. We'll get to that later though. The very top piece is the eyepiece. This is literally just a series of lenses. There's actually 2 lenses in here. It's called the eyepiece because that's the part you look through. That sits in the barrel, or the eyepiece tube as it's sometimes known. The purpose of this is simply to connect the objective lens, which is your main source of magnification, to the eyepiece. So as you can see, if you look down there, you're not going to see much. unless you put the eyepiece on, and okay, it's not in focus, but you can now see the aperture on the stage there. Coming back to the objective lenses, there's 3 of them in this case, and they're actually mounted on a rotating turret. This allows you to select which objective lens you want for a particular application. It should be noted that you always start at the lowest possible magnification and then work your way up. So in this case, I have a 4 times, 10 times and 40 times magnification. The eyepiece is also a 10 times magnification, so just now it's at 40, a total of 40 times magnification, 4 times 10. This particular microscope goes up to 400 times magnification. Next up, you have the stage where your sample sits. I have a couple of pre-prepared samples. I'll just use this one here. I'm going to hold it in place using these stage clamps, which are exactly what you think they are, clamps that go on the stage. Once you've got this done, you would then focus. You'd rotate the focus knob and bring your sample slowly into a sharp focus. I'll demonstrate this later once I've got the digital sensor in place in the eyepiece and we can have a look through it. There's one thing we're forgetting though, and that is underneath here there is a mirror. The purpose of this is to reflect light through the sample. So I'm just going to use a regular lamp here, and you'd look through the eyepiece. Sometimes with a bit of paper I don't bother with that. You look through the eyepiece and move the mirror until you've got enough light passing through the sample that you're able to see it correctly. And that's pretty much all there is to it. There's one small thing I'm going to mention here since it is a feature of this particular microscope. If you look at I'm going to turn that off, it's swamping out the vision. Underneath here there is a rotating ring with different sizes of aperture. The purpose of this is to control the pattern of light, and just like in a regular camera, by changing the aperture you change the depth of field of your sample. In this case, the depth of field is usually measured in fractions of a millimetre, but it's a depth of field nonetheless, and when you're dealing with something that's fractions of a millimetre wide, it can become quite useful to be able to change that. Next up, we have microscope slides. I said that funny. You have microscope slides and cover slips. Now, ultimately, the purpose of these is to allow you to get a clean sample mounted in such a way that you can look through it and examine it. However, there's several different ways that you'll need in order to prepare a sample, depending on what it is you're actually doing. For instance, I have here it's kind of gross actually, I'm going to get a new one, back in a minute. This is quite a large sample. This is a plant. I don't know what plant it is, it grows out my backyard. There are various different ways to prepare a sample, which was the point I was getting at. In this case, I want to look at a couple of very large solid samples. I want to look at a leaf bud, a leaf, and a cross-section of the stem. These are all dry samples, in that I will be sitting them directly onto the slide. In order to prevent some of them drying out, the thinner samples in particular, I'm going to place a cover slide on it, but I'm not going to add any fluids to it. So this is a very simple slide preparation. First of all, yes, this cutting board is filthy. No, I don't care. I'm going to make a quick, sharp incision through the stem, which should hopefully allow me to... I just done that the wrong side. Let's try that again. I'm going to make a quick sharp incision, and then I'm going to make a smaller incision right beside that one. This assures that I've got a wee tiny sample. And again, as per dry mount procedure, we just sit that on there, and that's us. Job done. We can now look at the cross section. As I said, I also wanted a section of leaf, so I'm just going to very carefully cut. In fact, you know what? I want the leaf end on, like this. I'm just going to try and very carefully cut that in half by holding one end, taking a very thin sliver off it. That didn't work. Usually you'd use a microscope to do something like this. But again, I'm just going to take that tiny wee sample and drop it on. You'd not necessarily always do multiple samples per slide like this. In fact, I prefer to do one slide per sample, but I don't want to have to clean more microscope slides because cleaning them is a bit of a nuisance. Finally, we have this leaf bud. Again, just going to cut it. In fact, I'm going to cut this one lengthwise. In fact, I'm going to take a new one. I'm going to cut that lengthwise. Dear. Again, be very careful. These scalpels are sharp. Now you'll notice that this is a very thick sample. Let's spread these out a bit. A lot of these are very thick samples, and I'm not necessarily going to be able to get a proper view on them. With this big sample in the centre, I'm going to try and flatten out a bit by adding a cover slip. I'm not terribly confident that's going to do much. I should really have put this in a microscope and cut it correctly in such a way that you'd be able to get a very thin sample that the light will quite happily pass through. So for bulky things, this is all you need to do to look at it under the microscope. Put that to the side. I need another slide out. The next dry sample we're going to do is a smear. Well, sorry, it's not a smear. It's actually a type of wet sample. It's a peculiar one that sits halfway between dry mount and wet mount. We're going to take a liquid sample and we're just going to smear it across. In this case, I'm going to use cheek cells. For this, you take a little cotton bud and rub it up and down the inside of your cheek. That's it. Now to look at this sample, all you need to do is take your swab, and I'm going to just make a little S-shape motion back and forth to get the sample thinner and thinner and thinner as I go across. Now I'm not sure if you can see that. Let's see if we can get the light to reflect off it. Ah, yeah, you can just about see it. You can see the where the sample starts and gets thinner as I go further along. This is just because we're smearing it out. So that pretty much covers wet mounts. Dry mounts, begging your pardon. They're fairly simple. They're very easy to do. You just take the sample and drop it onto the slide, optionally putting a cover slip over the top of it. The next sample type we're going to do is the wet mount. Again, this is very simple stuff. For this sample, I've chosen some river water that I got from the local river. I got this particular sample from a particularly mouldy spot that was slightly submerged. I believe it was a black mould of some sort, probably. The very basic principle of the wet mount slide is that you put a drop of it on your slide. You don't need much. That's it. Now, while you can leave it... Oop, that's not centred. While you can leave it like that, you'll see that's actually that's a very thick sample. So what I'm going to do is I'm going to very carefully drop a cover slide onto it. Sorry, I'm not going to drop a cover slide, I'm going to lower a cover slide onto it. The purpose of this is to flatten out the sample, prevent it drying out, spread it out, but the reason that I'm lowering it slowly onto the sample is to avoid trapping air bubbles underneath it. Because nobody likes an air bubble. There you go. That's one wet slide prepared. I'm then going to do the same thing, only I'm going to use a raised cover slip. I'm actually going to do two of these, because I feel like it. These are specialist raised slides. They've got a little metal annulus. No, an annulus is a gap. They've got a little metal ring on them. And there's two ways that you can do this. You can either use this to suspend a sample, in which case you put a very, very small sample on the cover slip, turn it upside down, and suspend it above your slide so that it's hanging. This is called a suspended droplet or hanging drop method. It's very useful if you want to measure motility. In the previous case with the wet mount, we've smooshed our sample very low. We've sandwiched our sample between glass, which is not particularly useful if you want to see the things move, because you'll have squashed them. Conversely, I'm not sure how much sample I've actually got in this, so I'm going to take a very large sample between a cover slip just quickly drop in here. Now, as you can see, this is a very large sample, especially compared to the others. The reason for this is that I want to sandwich a thick layer, there we go, between the bottom of the slide and the top of the slide. Now don't worry if you don't have this particular type of slide with the metal ring on it. You can actually do this by taking a cover slip and putting 4 little dags of blue tack on the corners of the cover slip, and then doing that and using the blue tack to raise the cover slip instead of the regular way. And that covers the basic methods of preparing cells. Although I am going to say that the smear method is technically a method unto itself. It can be considered a dry mount, it can be considered a wet mount. Most people call it a smear. Right, so let's take you through the absolute basics of using the microscope. I'm going to take off the eyepiece and replace it with the digital sensor. The sensor is just a little CMOS sensor, exactly the same as you'd find in a camera, only it's mounted behind a little lens. Now some people prefer to put a bit of paper underneath this, and if you look down here you can actually see where I am focusing the light. Now, the very first thing we do is we take our microscope slide, center it, again make sure you're in the lowest position, the lowest objective lens. Just going to move the camera around until I've centered the sample. Starting from the top, you slowly rotate the focus knob until your sample comes clearly into focus. It's a bit difficult on this because the frame rate's not terribly amazing. Come on, where are we? There we go. That's just about us. Unfortunately, because of the nature of this dry mount being a very thick sample, it's very difficult to make out details in our sample. You can see the edges where it's been torn by the blade, but it's very difficult to see the parts of the sample itself. Looking at this cross section of the leaf, and again it's again you can't see through it. The other samples will hopefully be a bit better in this regard. Now it's as you can see, this sample is huge. It's not you're not going to be able to see much using transmission light microscopy. So this is where reflective light microscopy comes in. What I'm going to do is I'm just going to take a torch and shine it on the sample. You can make out a lot more detail in the sample now. The reason for this is that we're no longer relying on the light from passing through the sample. Now you can start to see, you can start to make out the individual parts of the sample. Don't mind these, I should probably point out, these dots here. That's actually oils and dirt on the cover slide because I've apparently not cleaned it properly. My bad. But looking at the inside of the sample, I wish I would stop auto white balancing. You can now see the damaged cells where I cut it. At the edge here you can see the edge of the cells where I've actually cut through them. And again, let's look at the other sample. This is the cross section of the leaf. Oops, the audio has actually cut out of the mic here, so I'm going to have to overdub the footage in post. Here we have the cross-section of the stem. In the centre you can see the phloem, which is primarily for sugar transport. Surrounding that is the xylem, which is for water transport. And surrounding that is the bark, although I can't remember the technical name for it, which is just for protection. This is just a quick demonstration of the difference between transmission of light and reflected light. Over here we actually have a group of fungal cells. These are known as hyphae. These are used primarily for feeding. You can see a central branch in the... so you can see several. You can see the structure is very branching. It's got a very large surface area. This is usually used for feeding. Fungi are saponotrophs in that they saponify their food. The hyphae extend towards the food, as you can see here. You can actually see it's grabbed onto the substrate here. Once it's grabbed the substrate, it digests it with enzymes and then reabsorbs it. The branching nature is very important for getting a very large surface area for the least amount of material put into it. Right, so I think that will do us for just now, folks. If you've enjoyed the video, why not leave a like? If you want to talk about it, or discuss microscopy, or give me any suggestions, feel free to leave a comment. If you want to see more like this, why not subscribe? This has been the Compendium of Mad Science. I'm your resident mad scientist, Callum P. Robertson. Take care, folks.